Welcome to Chapter 5. So in this chapter we're going to be talking about a couple different biotechnological techniques that are used today quite frequently in research labs. So really all biological research will deal with at least some of these type of technologies, and you've probably heard of some of them. So first up, biotechnology is the process of using technology to advance biological research. So scientists will use several different techniques to manipulate DNA. It goes into basically being able to decode DNA, manipulate it, identify the source of the DNA, all different kinds of things. So scientists will use different chemicals, computers, and oftentimes bacteria to work with DNA. We'll see in a later section why the bacteria are so useful for this, but they're not always as bad as people think they are. So scientists will use these tools both in genetics research and in just simple biotechnology. Biotechnology really kind of got its start because they discovered how to cut DNA in specific places. May not seem all that significant, but when you have several billion base pairs, okay, you don't really know what you're looking at. There's too much information to actually look at unless you can cut it up into smaller pieces. So, they did this through the use of what are known as restriction enzymes. So restriction enzymes will cut DNA in a very specific way. So the restriction enzyme will basically attach to the DNA and kind of scan down it, looking for a very specific sequence. Once it finds that sequence, it'll kind of latch onto it and then cut it either before the sequence, in the middle of it, or at the end. Okay? And it's a very predictable way that it cuts. Okay, so this is showing a couple different restriction enzyme sites on this piece of DNA here. So they act as basically they're called molecular scissors. They're very, very precise, and they will just go in and cut the DNA. So the location in the DNA where these restriction enzymes will cut are called restriction sites. So actually the name finally makes sense here. And these enzymes actually come from various types of bacteria. The restriction enzymes were kind of the bacteria's safety system, like their immune system in a way. If any foreign DNA came into the cell, the bacteria cell would release its restriction enzymes and have them go cut up that DNA so that the foreign DNA could no longer be used. So of course, once scientists found them, they realized the use for them and started taking them out of bacteria and purifying them. So this allows scientists to relatively easily manipulate genes and study the effects of different genes, different mutations, things like that. Okay. It also means that if you're studying a particular gene, you can isolate that gene. You don't have to study the entire organism. So, depending on which restriction enzyme you're working with, they will cut the DNA in a couple different ways. So, each of them has their own unique restriction site sequence. Some of them can be relatively long, some of them can be short sequences. So even if you have the same piece of DNA, if you use two different restriction enzymes, you're going to get a different cut pattern. So this is just showing the first enzyme has 3 restriction sites, so it cuts the DNA into 4 pieces. For the second restriction enzyme, it actually has 6 restriction sites, so it cuts it into 7 different fragments. Okay. So some restriction enzymes will cut straight across the DNA, so there is no zigzag kind of pattern. It's just right across the 2 strands of DNA. and that leaves what are known as blunt ends. So they are very short kind of ends, they're very straight across. You also can have what are known as sticky ends. So these are your staggered, your zigzag type cuts. If you look here, okay, let me get my laser pointer. Okay, so this restriction enzyme has two sites here. And so enzyme will come in, find its restriction site, and cut the DNA. So this TAC1, is how you say that, TAC1, cuts the DNA in a sticky end pattern. Okay, so its sequence that it looks for is TCGA or AGCT, okay, which is just the backwards form. Okay, so it attaches to the DNA and it cuts in this zigzag pattern to create its sticky ends. Okay, and this actually has two sites, and you can see here, so it cuts it in two places, meaning that you have a longer piece of DNA here, your short little piece right here, and then another really long piece off to the end here. So this may not seem like a whole lot, okay, being able to cut DNA like this, but when they started trying to sequence the human genome and other genomes, there was just too much DNA to actually run through the machine, and the machine would start losing accuracy the longer the piece of DNA was. So being able to actually cut the DNA into pieces was actually really useful for that. Then they could just feed short little pieces of DNA into their sequencing machines and get back the exact nucleotide sequence of that piece of DNA. And then they could go from there with the different research they were doing. So what they were able to do was create what are known as restriction maps. So restriction maps will show the length of the different DNA fragments in that sample. Generally, this is done using what's known as gel electrophoresis. Yes, that is a really weird word. You get used to it after a while, and then it kind of almost becomes fun to say. But gel electrophoresis is used to separate DNA fragments based on their size. So you may have seen something like this, especially in like crime scene shows. They like to kind of insinuate that you can make one of these in like 5 seconds. It usually takes at least an hour, if not longer, depending on how big of a gel electrophoresis you're doing and a bunch of different things. But basically you'll get these bands, these, in this case, the glowing bands here, okay? Now, they only glow because they have a fluorescent tag on them, so it's been treated in a certain way to make the DNA fragments light up. And you'll see that this on the side here, this is a standard, so it's the control for this type of experiment. So they will run what's known as a DNA ladder, which has very specific DNA fragments in it, with which each have their very specific sizes. So then if, say, these two fragments here are in line with each other, you know that this fragment from your sample, your unknown sample, is almost the identical size as this one from your ladder. Okay, so you can tell what the size of this fragment is based on its relation to the ladder here. Okay. All right. So, walking through the process of gel electrophoresis. First up, you have to cut your DNA sample with restriction enzymes. Generally, they will use multiple different restriction enzymes. Okay, it really depends on how small fragments they want and the DNA that they're working with. And then they'll load these fragments into the gel. So I'll actually go back and show you here. So this is the gel here. It's usually maybe 1/4 of an inch thick, okay? And it has these little wells at the top, right? So they're just indentations in the gel. And you can load your DNA sample into these little wells, okay? And then You put this gel in a buffer solution, so it's a liquid that will conduct current, and you plug it up into your electrophoresis machine and turn the current on. The current will actually pull the DNA fragments through the gel. Okay? DNA has a negative charge, so it will actually move towards the positive end. Okay, so the electrical current, it will move the DNA towards the positive end. Now there's resistance in the gel. Okay, it's like if you were trying to force your way through one of those ball pits that kids always love to get in because their parents can't find them. Imagine the kid moving through the ball pit, and he's small enough that he can like wiggle through all the different spaces between the balls. But then when the parent tries to go after him, you know, the balls are kind of pressing up against him, not enough space to really move through it easily. Okay? Same kind of concept with the DNA fragments. Smaller fragments can kind of wiggle through the spaces in the gel, and they move faster and travel farther than larger fragments, which kind of get stuck in the gel. Okay? And so they have to move a lot slower. And so fragments of different sizes appear as those bands on the gel. And there are a couple different ways that they can dye them to get those bands to show up. So a restriction map shows the lengths of the DNA fragments between the restriction sites. Okay, so this is an example of a restriction map on a gel electrophoresis. Okay, so you have your negative end at the top and then the positive end at the bottom. So your DNA started up here and then moved downward towards the positive end. Okay, so this is the DNA sample you started with. You cut it with some restriction enzymes and ended up getting 4 fragments from that. When you run all of those fragments on a gel, you will get your longest fragment up at the top, and then it will continue downwards with the shortest fragment moving the furthest from the starting point. So, restriction maps are really useful to see size, and we'll see exactly why it's all that useful for that, but they only indicate size. They don't actually indicate DNA sequence. Okay, so that's a key difference. You actually have to use a different machine. It's called a Sanger sequencer in order to look at DNA sequence. So it is useful in genetic engineering, because if you are trying to insert a gene into a certain area, you'll see a massive size difference if you were successful in inserting that gene. Okay. It's also used to study mutations, because oftentimes mutations will cause some kind of difference in your restriction map. Okay, so you'll be able to see a size difference in the fragments. Okay, so we're actually going to pause here and then continue in the next video.